Okay, so I want to go over this press release that comes out of UC Davis talking about using CRISPR technology, which is a form of genetic engineering, to insert the SRY gene into a fetus, and in this particular case, a bovine fetus. So, what is the SRY gene? So, the SRY gene encodes for the SRY protein, which has now been named the testis determining factor. So, the testis determining factor is a DNA binding protein, so hence the name factor. So factors are frequently proteins or signals that can bind directly to the DNA. And by binding directly to the DNA, they can upregulate or downregulate the transcription or in other words, the activation of a gene. And so if you have certain genes that have the binding site for this SRY protein, those genes can then be upregulated or downregulated. So, in the case of most mammals, and I believe all mammals, but there might be a few exceptions, the sexual dimorphism of those animals is determined by their X and Y chromosomes. And if there is a Y chromosome, which is where this testis determining factor is encoded on, then that will be that particular factor is going to be translated, and then as this translated factor exists, it's then going to change the pathway or change the genes that are going to be expressed to determine the male pathway of that particular organism. So, and that's just what's happening. Obviously, there's going to be a lot of variation in biology as far as what genes are upregulated. Some will be more upregulated in some people versus others, but for the most part, that is the bifurcation of the sexual dimorphism right in that location. So this means that theoretically, this simplifies the process of genetically engineering males versus females as far as changing the outcome of what a embryo or what a fetus is going to do. So if it had to be an entire chromosome or an entire set of genes that are distributed throughout the entire genome that you have to add into a cell to make it be male versus female, that would be very complicated to do. However, if it's a single gene which then interacts or causes all those other proteins and transcription factors to change, then you basically can just do one small gene insertion or mutation, and you can basically redirect the path that the growing, developing organism is going to take. So with this, I'll just continue. So scientists at the University of California, Davis have successfully produced a bull calf named Cosmo, who was genome edited as an embryo so that he'll produce more male offspring. Okay. So the research was presented in a poster on July 23rd at the American Society of Animal Science meeting. So this has not been peer-reviewed yet. This is not something that really has gone through. There's no paper that is written on this explicitly, but frequently laboratories will have poster presentations for different sorts of symposiums or conferences that they have, and sometimes people will have posters on the research that they are doing, but has not been released yet to the public. So this is something that will probably end up as a paper later. So using the genome editing technology CRISPR, researchers can make targeted cuts to the genome or insert useful genes, which is called a gene knock-in. So gene knock-ins would be the opposite of a gene knock-out, where you remove or you basically silence a gene using some form of genetic engineering. So or I shouldn't say genetic engineering, there are multiple ways to do it, but in this particular methodology, we're talking about genetic engineering. So in this case, scientists successfully inserted or knocked in the cattle SRY gene. And remember, the SRY gene is this testis determining factor. So this is the gene that is responsible for initiating male development into a bovine embryo. So they put that into a bovine embryo. So it's the first demonstration of a targeted gene knock-in for a large sequence of DNA through embryo-mediated genome editing in cattle. So I said that, yes, it's a small gene, right? But actually, genes are pretty large as far as genetic engineering into a living organism. We don't really do that much. Usually it's like point mutations or just small little oligonucleotides that we add in or subtract from these organisms, doing an entire gene is actually a pretty large feat in of itself. Anyways, we anticipate Cosmos' offspring that inherit this SRY gene will grow and look like males, regardless of whether they inherit a Y chromosome. So, somewhere, I don't know where, they decide to do this, but using this CRISPR technology, they have decided a particular chromosome that, or a particular nucleotide sequence where they want to do a cleavage and then do the insert of this gene. Now, granted, I don't know how many genes or how many cells ended up getting this mutation. That's one of the problems with dealing with organisms that have already been fertilized, is that there are multiple cells, and sometimes all the cells don't take up the genetic engineering. And since there's no paper, I can't look on it to see what methodologies they exactly used. But what we're saying here is, with a recently fertilized embryo, they were able to genetically engineer this bovine to express the SRY gene, or have the SRY gene on a chromosome that is not the Y chromosome. So whichever chromosomes they added to the organism, if the offspring inherits any one of those chromosomes from the father that has the SRY gene, then that offspring is also most likely going to develop into a male, even if that male has two X chromosomes. Okay, so let's continue. So the reason why they're saying that this is the reason why, if you have more males, you have more beef, and that the male cattle are the bulls are basically where we get most of our meat. Now, granted, I think some restaurants use old milk cattle as a way, or basically cows. They use old cows that are no longer productive for milk as a source of meat, which is very cheap, but that's, lower level, I should say, less quality meat. So most quality meat comes from bulls. Anyways, Van Enneman says that part of the motivation to produce more male cattle is that male cattle are about 15% more efficient at converting feed into weight gain. They are more fuel efficient than females. Additionally, they tend to be processed at a heavier weight, so you get more bang for your buck if you have a male versus a female cow. And there's probably a lot of reasons for that, but female cows probably have different organs and different, what would the word be, different metabolic processes that they have been evolved to have, most likely for the development of another living creature inside of them, right? So anyways, it could also be a win for the environment. With fewer cattle needed to produce the same amount of beef, ranchers could produce some females as replacements and direct a higher proportion of male cattle for the market, said Joey Owen, a postdoctoral researcher in animal science who is leading the project with Van Enneman. Sorry, if I'm mispronouncing the name. So, an arduous journey. So the SRY gene was inserted into bovine chromosome 17. So they answered my question, which is a genomic safe harbor site. So perhaps they chose chromosome 17 because it is highly, it could be a chromosome that is very often expressed. Yeah, I wouldn't be able to tell you exactly why. Anyways, that ensures the genetic elements function predictably and don't disrupt the expression or regulation of adjacent genes. So chromosome 17 was chosen after unsuccessful attempts to knock in the gene on the X chromosome, which could have resulted in a bull that produced only male offspring. Cosmo is expected to produce 75% male offspring, the normal 50% XY animals, and another 25% XX animals that inherit the SRY gene. So they only, I guess what I'm a little confused at is, if they chose chromosome 17, how would they target just one of the chromosomes and not both of them, if that makes sense? So you have the 23 pairs of chromosomes, right? So how did they target one specific one? Because if it's in both, of his chromosome 17s, then it wouldn't matter which child, or it wouldn't matter which chromosome the offspring ended up getting in their, from the sperm. So theoretically, both of all their children will either be male because they have the XY and this additional SRY gene, or they will all be male because they are XX and then have this additional SRY gene. I'm a little confused as to why they say 75-25, unless it was explicitly just one of the chromosomes. So I'll have to look at the math just to get my head straight on it. Anyways, it took 2.5 years to develop the method to insert a gene into the developing embryo, and 2 years to successfully establish a pregnancy, said Owen. But in April of 2020, a healthy 110-pound male calf was born, and this has been a real labor of love. So she said this is just the beginning of the research, and Cosmo will reach sexual maturity in a year, and he will be bred to study if inheriting the SRY gene in chromosome 17 is sufficient to trigger the male developmental pathway in XX embryos, and result in offspring that will grow and look like males. As the Food and Drug Administration regulates gene editing of animals as if they were drugs, Cosmo and his offspring will not enter the food supply. So that is good that we are going through testing before we allow people to eat this, because let's say there's some sort of genetic deformity that happens, or let's say there could be the possibility that there is a huge dysregulation of genes, and then that results in different metabolites that might not be as healthy to eat. I mean, I'm just spitballing right now, but those are some possible things that we need to worry, and only after doing the FDA trials, but only after having the FDA look to see and do the risk-benefit analyses would this end up going onto the market. So, yeah, I'm still a little confused. I can't wait for them to actually get a paper out so that I can look over the math myself. But I'm confused because if it is on only one chromosome 17, then I can see the 75-25 split. But it's probably going to be on both of them. So I'm just a little confused. Now, granted, maybe they're expecting that only that the cells that do get genetically engineered, there's only enough material that they inserted into the embryo to successfully, or by chance, only about half of the chromosome 17s are going to end up with this additional SRY gene. That is possible. Now that I think of it, that might be the rationale that they're going with. They're thinking not all the cells are even going to get genetically engineered, and the ones that do get genetically engineered, sometimes only one of the chromosomes will be edited, and sometimes both of the chromosomes will be edited, and so they're probably just saying about half, right? And so if it's about half, and if that translates into the sperm cells as well, then they could probably make the argument, yeah, about 75% of the offspring are going to be male. Okay, so that's interesting. So part of me is also interested in how does this relate to human research, because there's probably a lot of people out here who would be interested in doing genetic engineering on themselves to improve, or not improve, but change characteristics about themselves that they don't necessarily like. I mean, we do that already with plastic surgery. We do that already with hormone replacement therapies. We do that already with, a lot of things, right? So would genetic engineering, since we are practicing it so much on cattle and our food production, there's probably going to be a very quick, what would the word be, basically a snap into when we go from doing the research on animals to doing the research on people who want to have some sort of therapeutic use. So that's going to be the first thing. Therapeutic use is going to be probably where a lot of this CRISPR genetic engineering goes first, and then eventually it's going to move more towards vanity projects of the ultra wealthy, right? So probably the ultra wealthy are going to look at this genetic engineering and see all the positive results that can happen when it's used therapeutically, and a lot of them will probably decide, oh, maybe I want to expand on this just for my general well-being, right? And so it'll first become something that the very wealthy people will invest in, and basically do a lot of the they'll do the funding for the product development, I guess would be the best way to describe it, and then eventually it'll go from being theoretically, possibly it'll go from just being for the ultra rich to being something that the average person might want to participate in, and eventually when you have, what would the word be? When you have enough people, there's a specific economic term, and I can't think of it at the top of my head, but there's a term that if enough people want to have a particular product, and there's enough of that product to go around, then the price will actually, plummet, and it'll become more affordable and accessible for everyone to use. So, I mean, a lot of rabbit holes, right? A lot of ethical and moral, I guess, complaints could arise out of this. Why can't we just love ourselves for the way that we were, born and conceived? But this is going to be the reality for a lot of people, is genetic engineering will play a role in changing characteristics about ourselves that we don't like. So yeah, let's see where this goes. Let's see if their predictions are correct and that the animal will be able to have more male offspring than female offsprings. And if that is a success, then you might see in the future, men wanting to have more male offspring versus female offspring, which would not be, well, that wouldn't be out of the realm of possibility, because we do know that in all over the world, actually, I mean, even in the United States, there are a lot of people who want to have male offspring more so or more frequently than female offspring. And so there has been a big concern about, selective abortions, and yeah, I mean, that's the big thing, right? There's a lot of selectivity that goes on because there is a differential in the outcomes of the family and outcomes of the life of the offspring, depending on what their gender is, right? So, and depending on what their sex is as well. So there has been selective abortions in the past to choose what sex I want my child to be. So would we end up seeing these types of technologies be utilized to have more female offspring, more male offspring, or would we just move straight into genetically engineering the embryos themselves? So a lot of this, is difficult to grapple with, right? And yeah, who knows? And granted, you could have the opposite thing as well, where people possibly want to deactivate their Sry gene so that they only have female offspring. But granted, that would probably be a much more damaging. Removing genes, I believe, has much more dire consequences for the health of the offspring. So be wary of that. So adding one gene versus removing that gene is not going to have, it's not going to be linear. It's not going to be, what would the word be? They might not be proportional to each other, right? So one could be drastically worse than the other. So yeah, let's not go down that road. So with that, thanks for watching, and if you have any questions about any of the process here, I can't answer that much of them because there is no paper out, but you can ask them and I can do some searches to see probably what might be happening here. So thanks for watching, and hopefully I will see you tomorrow.